Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Hannah Collins. She's a Ph.D. student in Oceanography at University of Connecticut. It's in the Department of Marine Sciences. And we're going to talk about microplastics and how it affects clams and oysters and other creatures in the sea. So, Hannah, thanks for coming.
Hi there. Thanks so much for having me.
Yeah, no problem. Tell me a bit about your background and how you got into this area of research.
Yeah, so I got my bachelor's in biology from Gettysburg College back in 2016, which doesn't sound very long ago, but it feels pretty long ago to me. I spent some time working with freshwater mussels, actually, so kind of jumped right into researching aquatic systems. Yeah, I then took kind of a different job and ended up working at a salmon hatchery in Alaska for a couple of years, which was really interesting, really eye-opening, and kind of reignited my sort of childhood passion for the ocean, right? And so I knew I wanted to research the ocean, but I also really loved biology and really loved marine invertebrates, really loved mussels, oysters, clams, those kind of animals. So I started applying for graduate positions, and I ended up applying to do a master's position here at UConn with my advisor, Dr. Evan Ward. And so I graduated from the master's program back in the spring and kind of rolled right into doing a Ph.D. here as well. So yeah, I'm solidly in year 2 now of the Ph.D. program. Yeah.
Well, great. So what is your research about right now? What are you looking at in terms of microplastics?
Yeah. So right now I'm researching specifically the microbial communities that form on microplastics. So as microplastics get out into the ocean, they're colonized by a number of different bacterial and eukaryotic species. They're a substrate for things to attach to in the ocean, basically. And so there's a lot of growing evidence that community that forms on microplastics, especially kind of in the early stages when a piece of plastic first gets out into the ocean, it looks pretty different than communities that form on other types of substrates. So, a piece of seagrass that's floating out there is going to have different types of bacteria than a piece of plastic. And so what I'm investigating right now is how those different microbial communities on microplastics, specifically how they can affect, whether or not they affect the bivalves that I am studying. So the mussels specifically is what I'm looking at. And
so you're looking inside the mussels and everything to see if there's microplastics, or how are you evaluating this?
Yeah, so partly. So I have a coworker, another lab mate of mine, who looks at the types of microplastics that bivalves are ingesting. So they are suspension feeders, basically filter feeders, essentially. And they're filtering tons of microalgae out of the water, but as they're doing so, they are getting microplastics as well, because there's these really small plastic particles. So as a lab, we're studying which types of plastics they're ingesting, but also specifically, I'm looking at how those plastics with this associated microbial community, how those could affect potentially the gut microbiome of mussels and oysters. Because what's kind of interesting is we know that they do have actually a gut microbiome.
Yeah, well, I'm sure they do. I wonder if there's anyone that's studying how, when pearls are formed, what happens to the microbiome of these creatures and how does it change, and et cetera. But I know it's probably a different subject. So.
Oh, that's an interesting question. Yeah, so when the pearls are formed, it's right next to the shell, the mantle of the organism, so kind of like the skin. That's what's secreting that material that forms the shell. So it'd be interesting to look at the bacterial communities that are on that mantle tissue.
So what are you seeing in all these bivalves? Like, is there a lot of microplastics? And I don't know. What do you think the circumstances are in which they take them in?
Yeah, so actually what we're seeing is there's not a lot of microplastics that kind of accumulate in bivalves. So bivalves are actually selective feeders, so they don't eat every particle that they capture. So they have gills that are basically they use them for getting oxygen, but they also use them to capture particles out of the water. And they can they on those gills, and then also they have special organs that can select different particles that they either want to ingest or don't want to ingest. So if there's a microplastic that's, for example, too big, they're not even going to ingest it. So that's good news. But then also, just because of the way these organisms are so cleverly evolved to process tons and tons of particles per day, per hour, they have selection mechanisms on their gills, on special organs, and then also in their stomach. So actually what we're seeing is that even if they are ingesting some microplastics, they're basically pooping them out pretty quickly. So if you were to take an oyster from the ocean, it would probably have maybe one microplastic in its stomach, maybe two. So we're actually finding that they do a really good job of kind of clearing those particles pretty quickly.
Yeah, but I mean, each I don't know. I mean, so first of all, how do oysters and clams and all that, how do they breathe? I mean, they don't have gills like fish. So how do they take oxygen in, or how do they respirate?
Yeah, so they have, right. Sorry, go ahead. What's your question?
Oh, I was going to say, I just imagine them opening their shell and going in a way and taking stuff in. I'm probably not accurate, but they're probably with every respiration or breath or periodic opening of the shell, this garbage is coming in, and then they're maybe blowing it back out. But essentially, if that happens in and out, in and out, in and out, it's kind of as if it stayed there in a way.
Sure. Yeah. So what happens when they're respirating? So they do have gills. Obviously, they're not on the outside of their shell. They're on the inside of the organism. So it's a little different from fish. But those gills
I didn't know that.
Yeah, so those gills do serve as the organs of gas exchange. So they are getting oxygen through the gills. But bivalves, specifically the gills, have evolved to also capture particles. So it's they have two different functions. So they are getting the oxygen through their gills. They're also capturing the particles on their gills. And then on those gills, there are cilia that are basically creating water currents. So every time a mussel or an oyster is open, that's actually these cilia that are creating water currents, drawing particles in. Those particles are captured on the gill, and then they're basically transported to these organs of selection. They're called the labial palps. And so from there, they're either ingested if the animal, so to speak, chooses to ingest that particular particle, or they're egested, or they're rejected, and they're just spit back out. So yes, technically they're kind of moving in and out, but this happens with all types of particles, not just microplastic.
Yeah, but how much is the microplastic load that you've observed? And when there's a certain load, what does that do to its respiration? Mm-hmm. Does it cause it to do it faster or more deeply or less deeply, or maybe the quote-unquote expiration or exhalation of the respiration when it opens and closes? I don't know. Maybe that alters.
Yeah, so that's a good question. Actually, what we do find that, specifically we're talking about the ocean here, obviously, where these marine animals are living, we're actually finding maybe 1 to 10 microplastics per liter of water. So I'd say the load is actually a lot lower than maybe you would think just about thinking about plastic pollution in the ocean. So I'd say number one, the load is pretty low. And number two, so because these animals are, they're designed to process tons and tons of particles as they're feeding, they have mechanisms to kind of shut down or reduce the amount they're feeding when there's just too much in the water in general. So not necessarily microplastics, but essentially what they'll do is they'll just kind of stop feeding for, a period of time until they sense that the particle load in the water is lower and they can actually process particles. To your question about respiration, I'm not entirely positive. I don't study a whole lot of respiration, but some of the things that mostly affect respiration are temperature, for example. So they'll respirate more, they'll respire more when the temperature is higher.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
But
What
I
about
don't
the
think
movement
that-
of the shell? Is that at all coordinated with respiration? And does the shells? I imagine them, I guess, opening and closing their shells, or do they kind of sit partially open and then they close at night? Like, what happens to the shells, and how's the action of that affect?
Yeah, so they'll have to open their shells to respire and to pull in water with oxygen. That's typically associated. They'll either be like a little open and they'll draw some water in and kind of they can pull oxygen out of the water. When they're open, really open and feeding, they're also getting a lot of water in. So it's kind of correlated with that. Yeah, there's a large body of research about respiration in bivalves. I wouldn't call myself necessarily an expert on respiration specifically, so I don't know how well I can answer that question for you.
Okay. Well, what questions are you trying to answer with your research right now specifically?
Yeah, so I'm trying to look at how the bacterial communities on microplastics, whether or not they're affecting bivalve gut communities. So I did some work on that with my master's thesis. And so long story short, they're not really affecting the bivalve gut community, which is pretty interesting. And so kind of moving off of that research, I'm now looking into whether or not you could use mussels as kind of a biological solution to microplastics. So because we know that they process large quantities of water, we know that they capture particles and they process them and either, kind of poop them out or reject them. When they reject them, they package them into mucus and those biodeposits sink to the bottom. So we know they can kind of remove plastics from the environment. So could we potentially use them as a biological solution to the microplastic problem and use them to remove them from the water? So that's kind of the direction my research is going. Yeah.
But they package them up and spit them out, right?
Yeah, they do. And so
when they do that, do they agglomerate, or they get stuck to the ground, or does it immobilize them?
Yeah. Yeah. So when they package them up, so that's either in the form of the feces, so as they, the particles move through their digestive system and get pooped out essentially, or if they are rejecting a particular particle, they kind of package up a group of particles in mucus and spit that out. And that's called pseudofeces or fake feces. And both the pseudofeces and the feces are nice, big, dense particle, groups of particles that are going to sink to the bottom and kind of sit on the sediment, basically. So it is going to remove them from the water itself.
Okay. So you think maybe by cultivating, let's say, a whole bunch of oysters in a place where there's a lot of microplastics, it might take a significant amount of them out of the water in the form of like goo on the bottom of the ocean?
Goo is a good way to put it. Yeah, so that's something we're exploring right now. We're actually looking at some freshwater mussels as well, seeing if you could do that in a freshwater system. They function the same way. They still select a ton of particles and package them up in these really dense biodeposits. So that's what we're investigating, the feasibility of doing that right now. Yeah.
How do these creatures suck water in and spit it out? Is it coordinated against the opening and closing of the shell? Or do they have other muscles or type of suction and blowing they can do?
So it's actually cilia. So it's tiny, essentially, I guess maybe fibers is a good way to describe them, tiny cilia that are on their gills. So their gills are actually covered in these cilia and mucus. And so the cilia, by the way they beat, they create water currents to draw water in. And then the animal can also create the currents to push water out, or if they do close their shell, that'll also push water out of the shell.
Yeah, I just have the feeling that it's critical to know the position of the shell when they're performing different actions. Like if they, picturing an oyster with its shell almost closed. So let's say if the cilia are sweeping stuff out, it's going out through like a narrow place where the velocity would be very high because the shell's almost closed. And if it's open, I would think you'd have less impetus for stuff to be pushed out of the shell.
Yeah, so typically when you have a bivalve that's actively feeding, actively drawing in particles and, processing them, they're wide open. So they're going to be really open so that they can filter in as much water as possible. And if you have an oyster that's not very open, it's probably not very active, and it's not really actively feeding or actively creating those water currents.
But what determines whether they're going to be open or closed? Like, is it a diurnal variation in this, or how does the creature decide what to do?
Yeah, not so much. I'm not sure if there's a diurnal variation in that. Typically, the food concentration in the water. So if they can detect, through chemical cues or however we're little, then they can open up. So a lot of it is food concentration in the water that really stimulates them to feed.
Okay.
To open up and feed, yeah.
So in addition to them, sequestering some microplastics, I mean, when they do it, are they? So you said it's about 1 microplastic per liter of water, which is not a lot. So do they take in a bunch, kind of put them all together, and then spit them out, every so often, or are they spitting them out as fast as they can? A little bit of goop with 1 microplastic at a time.
Yeah, so it can kind of depend. So when a bivalve is feeding, it's processing this water kind of constantly, usually. Sometimes it'll stop and start back up again. But if it's processing, for 20, 30 minutes, it's processing this water. Some microplastics will get spit out individually. If they're really big, sometimes they'll just kind of pop off. Some get, kind of agglomerated into this ball of mucus that gets spit out. That happens pretty quickly. That can happen within minutes of a particle being captured. So it does happen pretty quickly. And then if it's actually ingesting the plastic, that the egestion of that, the feces can be produced within a few hours to up to maybe a day or so, depending on how long it takes to go through the digestive system. So it really depends. And what's kind of interesting is, we're investigating what determines the selection of microplastics, which ones they, so to speak, choose to ingest versus reject. And a lot of it is size dependent. So some of the really big microplastics, they'll just spit out right away, for example.
Well, what about when I believe it's a clam makes a pearl, and supposedly a grain of sand gets in there and more grains get in there, and the clam can't get rid of it. So it eventually forms a pearl. So why wouldn't that happen with microplastics? So they're like pearls with a microplastic in the middle of them, sadly, or what?
Yeah, I haven't necessarily heard of that. So, but I do know that when, oysters, for example, form pearls, that's, it's not inside their digestive system. So it's when, a piece of grit or sand kind of gets stuck in the tissue that's right on the shell, and it can't get rid of it. So it creates this layer of essentially what is shell material to kind of protect the tissue from that, that piece of irritating sand. So yeah, I don't know. Maybe it might be feasible that you could have a microplastic that could stick, and you could, I guess hypothetically, you could have a pearl formation from that. I haven't necessarily heard of that being found or being shown, but yeah. So again, that's kind of on the outside tissues, not necessarily in the stomach of the organism.
When it spits out microplastics with the goo around them, or in the feces form, how is that different from, again, pearl formation? Is the location of the aggregation of it, the goo, at the same point as where the pearl forms, or is it a different spot?
It's a different spot, yeah. So pearl formation can kind of be anywhere along the inside of the shell. It kind of just depends if something gets stuck there. When the animal is spitting out microplastics, either through rejected pseudofeces, which is that goo, essentially that mucousy kind of, rejected particles, or the feces, so that's coming out through the intestine, through the anus, or right near the mouth is where the selection organs are. So there's very specific locations where pseudofeces and feces are produced, but with the pearls, it can kind of be anywhere along the inside of that shell. It just is, kind of random as to if a piece of grit gets stuck there.
Well, I guess the microplastics over a certain size are just, they don't go anywhere. They get caught by the cilia and swept out. But if they're quote unquote small enough, then they enter the digestive system, and then they're, processed another way.
Yeah. So the processing in the digestive gland is actually really intriguingly complicated. There's a ton of different tracts and tubules essentially in the digestive gland. So you kind of have a central stomach, but there's also smaller little ducts where if a particle is small enough, it'll get really digested. And so that can take up to, on the order of days for a particle to really be digested. But some particles that enter the stomach are too big to enter those tubules and those ducts, so they kind of move through within, more of on the order of hours, so to speak. So yeah, so I'd say, some of those big microplastics, even if they are ingested, they move through pretty quickly.
Do you see any where the microplastics have integrated into their shells? Like as the shell grows, does microplastics ever get caught in it and integrated into it?
Not that I've seen, no. So the shell is actually created by a tissue layer. So it's kind of secreted based on this, this matrix of organics and it'll secrete calcium carbonate to make the shell. I think it'd be pretty difficult for a microplastic to get stuck in there just based on the way that the shell is created. Yeah, but that's an intriguing question.
Do you see that any of the microplastics get into the flesh of these creatures? They can't get rid of it, obviously, and it just becomes a part of them?
So not microplastic. So I think that is actually getting into a question about nanoplastics, which are even smaller. So you definitely have situations where a microplastic can kind of get stuck to the outside of the tissue of the organism. They're kind of, everyone's eaten an oyster or a mussel, they're a little goopy, they're a little mucousy. So you can have particles that get stuck to the outside of them. But because you constantly have water coming in and out of the animal, those pretty much get washed away and they don't really get stuck. One potential concern that people have is that as plastics break down and they make microplastics, the further breakdown of that would potentially create nanoplastics. So these plastics might be small enough to actually get through into cells and get into the tissues of animals. So it's pretty hard to study nanoplastics, obviously, because they're so small. But that would potentially be a feasible concern if you have these really, really tiny plastics that could then, get through into the cells of the animal. But I haven't seen anything like that with microplastics.
Has anyone done tissue samples to see if there's, small bits of plastic in the flesh of the animals?
Yeah, so that's definitely something people have looked at. One of the issues with microplastics research is partly that microplastics are everywhere. So you have to have pretty good contamination protocols when you're trying to look at microplastics. And so, sometimes if you're looking at a tissue sample and you just happen to have like some atmospheric microplastics that drifted in, that can contaminate your sample. But you also, in order to accurately identify whether whatever you're looking at is actually a microplastic, you have to use spectroscopy to actually pinpoint the fact that it is a plastic. It's not just, a piece of sand or whatever it may be. And some of the problems associated, obviously not problems, but the drawbacks is that those types of spectroscopy systems can only identify plastic down to a certain size. So you do get a little size limited. So I'm not sure how well people have been able to look at nanoplastics in tissue specifically. I know that people have looked at microplastics. But again, with bivalves, because they are processing so many particles, and we know that they do ingest some microplastics, even if they kind of process them pretty quickly, you do find a couple in their tissues if you're looking at the tissues, but it's not huge numbers.
Okay, gotcha. In general, do these organisms attract more pollutants than, let's say, fish that you would eat, or less?
That's a good question. So, the way that they're getting at the microplastics is different from the way a fish would. So they are processing like huge volumes of water over the course of a day. So they are, processing microplastics that way. I'd say, a fish that's just swimming around and happens to be, getting some microplastics stuck in its gills or in its stomach or whatever, there may be fewer microplastics in a fish. Well, I shouldn't say that. So the fish may encounter fewer microplastics throughout the day, but I would expect that the fish might accumulate them more. I would expect that they might get stuck in the fish digestive system more easily because of the way bivalves are designed to constantly be processing all these really small particles through, whereas fish digestive systems aren't exactly set up the same way, right? They're not set up to process all of these tiny, tiny particles. Does that kind of answer the question?
Well, that's what I mean. Maybe fish are more amenable or susceptible to the buildup of these, and these creatures not as much because they can spit out a lot more.
Yeah, exactly. So I think, that's a good thing, right? Good for people who like to eat shellfish, right? They're not really accumulating all of these microplastics.
Yeah, no, hopefully not.
Yeah.
So what other questions are you trying to answer?
So yeah, I'm also looking more at the type of communities that make up their gut microbiome. I'm trying to see whether or not you could manipulate that gut microbiome, whether or not you could, put a particular bacteria in the water with a mussel, for example, for an extended period of time, see if that bacteria would colonize in the gut and become part of that gut community, or whether the gut community in bivalves is really specific. Only certain types of bacteria can live in that environment. So that's kind of the other half of my research.
Oh, well, what's interesting about the microbiome that you've observed now? What kind of bacteria are prevalent? I know they're heterogeneous, but what are some major ones that you maybe see a lot?
Yeah, so we're actually finding pretty frequently that we have mycoplasmas in there. So this is a particular genus of bacteria that they don't have a cell wall. So they live, kind of associated with the cells in the gut of bivalves. So they're pretty interesting little genus of bacteria. And we pretty much all the time find those when we're looking at the community in our mussels specifically. We've been doing a lot of work with mussels. And so what we also, we see that those are specifically associated with the mussels. But it's interesting because if you were to take a mussel right from the ocean and kind of dissect it out and use the techniques we use to look at, what does its gut microbiome look like, there are a number of species in there that we know are just like oceanic bacteria that are just in the water and they just get processed through the stomach. So what you have to do is kind of take the animal in and let it, keep it in some filtered seawater that doesn't have any bacteria in it, and then just let it kind of poop for a while and get everything out of its gut. And then once you do that, you can actually look at what we call the resident gut microbiome. So the community that's there all the time. It's not associated with the seawater. It's not associated with the food. It's the community that's actually living there.
So what are some of the differences that you've seen in the transient ones versus the ones that are always there?
Yeah, so a lot of the transient ones we see they're like cyanobacterias, which are really common members of just like a bacterial group that lives in the ocean. Some of the groups we see that are part of the transient community are photosynthetic bacteria, and these photosynthetic bacteria definitely couldn't be part of the resident community in mussels because there's no light in the stomach of mussels. They need light to photosynthesize. So we've seen some of those members, and then there's a number of bacterial, not even species, because sometimes it's hard to identify them at the species level, but groups of bacteria that we don't know a whole lot about, both in the transient and the resident community. So it's interesting, and it's an interesting branch of research because it's fairly new. People really have only been looking into, even human gut microbiomes in the last, maybe 10, 15, 20 years. And, marine invertebrate microbiomes, even less so. We've only got maybe 7 to 10 years worth of research that's just starting to kind of get at this. So there's a lot of questions we still have about these gut communities.
So again, once you identify the bacteria, then what? Are you going to do metagenomics, or what? What does it tell you besides, we see this one and that one?
Yeah, that's exactly the point. Is we can, at this point we're pretty good at identifying what species are there, but we haven't really done any of that metagenomics yet to say, okay, what are these species doing? There is a body of work in vertebrate species, obviously showing how intrinsically linked the gut microbiome is to health. And so our assumption is that, a lot of those mechanisms are at play with the invertebrates as well, but we just don't know yet. And so, hopefully down the line, doing some metagenomics, looking at some function of these bacterial species.
Well, very good. What's the best way for people to find out more about your research? Where can they go?
Yeah, so they can go to our lab website, which is, let me pull it up, but if you just Google UConn Marine Sciences Ward Lab, we have a lab page, we have a Facebook, I have a Twitter account too. That's, I post on when I have time, sometimes about research. My Twitter handle is at cloister, capital Oyster and capital Cloister. Yeah, I'd say those are kind of the best ways to keep up with what our lab is working on and our research.
Okay. Well, very good. Hannah, thanks so much for coming on the podcast. I appreciate it.
Thanks so much for having me on.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.